Okay, so what I'm going to talk about today is how you can derive emission factor data. Different approach from what we have seen in during the talks of this morning and yesterday. I will not spend much time on how we derive the emission data, but I will focus more on how we try to quantify the uncertainty associated with the estimate of the emission data. As we have seen this morning, there are many ways in which you can come up with emission factor, sorry, not emission data, and we have seen that you can use forest inventory data. That is the most common way. If your question is how much biomass you have in your country, you want one number with a confidence interval, forest inventory give you the best answer. The problem is that they don't give you much information on the spatial distribution of where the carbon is, right? So we have seen different ways to do that you can use strata, you can use land cover maps combined with forest inventory data, and what you usually do is that you pay attention in identify the strata, and in history in the forest inventory community, they've been doing this for a long time, and they know pretty well how to quantify the error associated with the estimate. Now, as we discussed, as many people pointed out, there are some limitation of this approach. For example, number of samples in each strata, the layer that you use for the strata, there is an uncertainty, an error associated with the land cover maps, for example, and so on. So what I would like to show you today is using a different approach that doesn't need to use strata, but what we do here is that we skip that phase and we try to build a relationship between forest inventory data or location on the ground where we actually know what the biomass is, and the information from the satellite, right? So we try to build an empirical model that give us the form and the shape of the relationship between the reflectance data, the information that we collect from the satellite, and what the biomass is on the ground. Now, as I was saying, right, if we do use a stratify and multiply approach, so we have the strata, we have the points, right, we need to make sure that we have enough samples to characterize the variance in each of the strata. We need to be careful about the errors in the thematic maps. You know, we have seen that each map has an error. And the larger the number of classes you have, probably the largest the error is in each class. And mainly, for my experience and my understanding, especially if we are trying to use this emission factor to characterize the CO2 emissions from land cover land use change, you do run into an issue because land cover are only partially able to describe the variability of the biomass. Here on the right are like the biomass distribution within one forest class. And this biomass distribution is shown as a function of airborne LiDAR data. So it's a sensor that provides you with sort of what we think today is the best sensor, the best way to get to the biomass. And as you can see, even if you have one class, the variance, the distribution of the biomass is pretty large. So if you want to quantify your emissions, well, you better know if the forest that you lost is here, it has 240 tons, or it has only 60 tons. While normal practice today is actually just using pretty much the average of the strata. And so depending if the disturbance is randomly spread in the strata or not, you need to make sure that you properly understand. But so the best way will actually be to be able to have matching data, right? Changes and the density of the biomass of that change. So that's cool there. So now, the problem of the empirical approach of building this relationship with the satellite is that you actually do need information from the ground. You need to have a data set that is going to tell you what the biomass is, because there is no satellite in space that is able to tell you what the biomass is. The satellites that we have now and that we will have, they're not able to give you a weight, right? Because after all, what we are after is to know the weight of the vegetation, okay? So, well, our project was actually covering the entire tropics, so it really was impossible to collect and acquire enough samples spread all over the tropics that would be representative of the sort of the variability of the biomass for the tropics. So what we did is that we relied on data from a sensor. It's a LiDAR sensor on board of an instrument that is called ISAT. ISAT was designed to measure the changes in thickness of ice, and so it has been stretched quite a bit to work on vegetation. But it does provide some good data that you can use to derive information on the biomass. I'm not going to talk about how the LiDAR works, but if you have questions, I will be happy to talk about that. So pretty much the idea here is, instead of using forest inventory data because we didn't have them, was to use biomass that we derive from the information of the LiDAR data. The LiDAR data is a sampling sensor that gives us information of an area of about 70 meters, okay, and it is a sampling sensor. So every 180 meters, it collects this sample of 75 meters. So what we thought is that, well, we are going to collect what we think the best representative data to calibrate a relationship that gives us estimate of biomass as a function of the information we derive from the LiDAR data. And here is how we did it. So we have our footprint of the LiDAR data. We design a field plot of 40 meters by 40 meters, and in this plot we measure all the trees with a DBH bigger than 5 centimeters, okay? Then we use allometric equation to convert the DBH to biomass, and we can see here on the top how actually what is called the waveform of the LiDAR data is changing as a function of the wood density of the plot that you have on the ground. As a result of this activity, we now have a regression model that is able to give us, for each location of the glass data, an estimate of biomass. And thanks to glass, we have like millions and millions of samples that you can consider them as a substitute for forest inventories. The difference is that those are estimates, right? Are derived from a model, and it's not like a forest inventory that you can consider it a measurement. This is an estimate. So there is an error associated with these values. But just to make it clear, so here is like how the sensor works. Every 185 meters, it collects the data, right? And so what we did is that we went across the tropic in some of those locations, right? So we measure the data, we know what the biomass is, and we test the model that then is able to estimate the biomass in all the locations of the glass data. So here on the right, So sort of black lines are showing the location of the glass data. So for each of these points, we sort of have an estimate of the biomass. We went across the tropics, 16, 17 countries, and of course, the glass data is not continuous. So there are gaps. As in the forest invention, you have location where you don't know anything. So in order to fill those gaps, we relied on the spectral data of the satellite, in this case of the MODIS data. Now here what you can see, it's a mosaic of a composite data over 2 years, because, you know, especially in the tropics, there are a lot of clouds. And MODIS is an optical sensor, so if there are clouds, you actually don't see the ground. But luckily, clouds move, right? So if you keep looking at the same place over and over and over, you will be able to get a clean observation. So we composite all this data over 2 years, and we came up with the data set. So now we have our estimate of biomass from LiDAR, from glass, and spectral data that will help us to fill the gaps, okay, for glass. So as a result, we came up with a biomass map for the tropics. Now, when we released this data and we published the paper, we were able to provide confidence interval of the estimate only at the national scale, okay? But what I was really trying to understand for me is what is the sort of the accuracy, the uncertainty at the pixel level. Because after all, we are saying that we are able to estimate a wall-to-wall map, right? So each unit will provide us with an estimate, but what do we know about how good that estimate is? So just to be clear again, here we have a three-step process. First of all, we collect the trees, we collect the dBH on the ground, and we use allometry to go from dBH to biomass. So allometry is an equation, right? And as we have seen, there is an error associated with that. Then we use this estimate in the LiDAR model, so we need to make sure to propagate the error from the allometry to the model of the LiDAR data. And the LiDAR data, again, it is a model, it provides estimates with an uncertainty. So then we need to make sure to have the total uncertainty of the LiDAR model propagated to the final step of the map. And it did take me some time to figure it out. And actually, I was not able to do it by myself, but as you saw at the beginning, our colleague, Luis, is a statistician at Boston University, and is sort of the main mind behind it. So now, I'm sorry, it's difficult to see this plot on the left here, but what the plot here on the left is showing at the three levels what the uncertainty is as a result of the allometry. So we are able to simulate the uncertainty of the estimate of biomass using Sharp's general equation, okay? Now, it is interesting to see that, you know, you do have an uncertainty at the tree level, but we don't really work at the tree level. What we work is at the plot level, right? So then what we did is that we aggregate those estimates at the plot level, and here on the right, you can see that for a random plot, you can see that your estimate can vary between 89 to 104 tons of biomass per hectare. And I was pretty happy with that. I will live with that. It's not a big error. Now, in this next plot, what I'm showing is, again, it's a different plot, and we have the estimate only from the allometry, right? And here on the right is the estimate from the allometry plus the error of the allometry and the glass model. So as you can see, there is a much larger spread, right? And below, again, is the allometry on the left in blue, and is the result of the glass estimate in red. As you can see, when you go from the allometry to the glass, well, you do increase your uncertainty. Now, again, that estimate was for one footprint of the glass data, so for an area of 75 meters. Now, we don't really work with 75 meters because what we want to characterize is the biomass within the MODIS pixel that is at 500 meters. So what it happens is that we have multiple shots, multiple points inside of the MODIS pixel. Okay? So as we were doing for the 3-level data that we aggregated at the plot, here we take the glass data and we aggregate them to the MODIS pixel. Okay? And so you can see again how the variance now, even based on glass, is not as bad as you can think it is if you look at the single shot. And again here, finally, then we propagate all the error of the allometry, of the glass, and of the last step that is generating the estimate for the pixel. And we can see here sort of taking a random pixel of MODIS where you don't have any glass data. This is just a location, a pixel where there are no information on the biomass. So you can estimate what the uncertainty is. Now you do that for all the pixels in the map, and you come up with the spatial distribution of the error. In this case, I'm just showing the percent error, so it's a percent error on the mean estimate of that pixel. And as you can see, you can go from 0 to up to 94% error in each pixel. Now, 94 sounds big, but if you do look at the frequency distribution of the percent error, you can see that the majority of the pixels have actually an error of 20%. Okay, and this is exactly for the area that you see on the left. Okay. Now we also tried out to decouple the 3 errors, right? We have an error from allometry, we have an error from the glass model, and we have an error from the MODIS data. And here on the bottom, I'm showing the error from the allometry. As you can see, that compared to the total error, sort of, the relative contribution to the total error of the allometry is actually quite small. Okay, it is about 7–10%. Now, if you are familiar with remote sensing data, we were lucky because we had 3 sources of errors, right? We had allometry, again, glass, and MODIS. So you can display these 3 sources of uncertainty as a RGB in a digital image processing software, right? And so what you will see is that if the error from glass and MODIS doesn't exist, your image will be all red, right? It will show up in red. If there is no allometry, no MODIS, only glass, it will show up in green. So now, when you look at this figure here, the color that you see is pretty much the color that has the highest relative contribution to the error. Okay? So what this map is showing, and it's sort of difficult to see, but error from allometry, we don't see much red. Okay? In the overall error, there is no much effect of the allometry on this map. What we see is that there is a significant error from glass, all the green, right? So the error from glass is dominating all the other errors. That is because it's showing up in green. And then we have some places that is bluish, is blue, and that is where the error associated with the MODIS estimate is dominating. Now, I do like a lot this type of information because it could help you to plan further activities. You know, you might go back to this and say, well, try to understand this. Why glass is so bad? Maybe I should just collect more field data to calibrate the model where I have the biggest error, instead of collecting data in places where the error is not big. I can sort of stratify my collection, my data field collection based on sort of the uncertainties. And now you can use the spatial distribution of the errors, and you can try to look at what is the error in different length over classes or in different strata. And as you can see, it is different depending, well, that one is impossible to see. It is difficult depending which forest type you are actually investigating. And as you can see that for the evergreen broadleaf type, the error is actually less than 20%. Less than 20%. In the mixed forest, trust me, was about 25%. Okay, it is higher. So this is interesting because if you want to estimate emissions, right, and you know that your forest loss is mainly from evergreen broadleaf forest to non-forest, well, you know what your sort of error could be. If your emissions are from the conversion of shrublands, well, you can see that your error again is different, is higher than the one from the evergreen forest. Just for curiosity, all those errors really are scale dependent. And here, just for curiosity, well, it's difficult to see again, but what I'm showing here is the estimate from our map at the national level for the tropics. So I took the total of the biomass for tropical countries that are covered by our map. And here on the left, I generated the estimate of the biomass following the IPCC guidelines with the default values that the IPCC is suggesting. And as you can see, there is, at the national level, there is almost a one-to-one line. Okay, so just to say that, you know, those estimates are really scale dependent, and you should always ask yourself, what is the use of your estimate? Right? Is it at the national level? Is it at which scale? Now, next step, with colleagues at the center, we are actually working on a specific map for Mexico, higher resolution, 30 meters. This time we are not using LiDAR data, glass data, so we will remove a source of uncertainty, and we will use the in-situ forest inventory data for the calibration. This is a preliminary map that has been completed, and my work in the next month should be to try to quantify the error associated with those estimates. Okay, following the methodology that we use for the MODIS-based work. So in summary, you know, we saw that at the plot level and for this sort of method to generate biomass estimates, allometry didn't really show up as the most important source of error. And this doesn't mean that allometry is not important. This to me means that the remote sensing data are so bad that pretty much allometry shows up as a small factor. Okay, so we do have a lot of work to do trying to improve the remote sensing data. And as I said, really it depends on the spatial scale at which you want to work. A recent study that we did trying to replace the LiDAR data, the glass data, with the airborne LiDAR data that they provide a much accurate estimate of the biomass is actually showing that you significantly reduce the error. Okay, so there is hope also because glass is a sensor that is not transmitting anymore, it's dead, so if you want to do a new recent study, you cannot do it. But airborne LiDAR data are becoming more and more common. We, as a part of a USAID project, MRED here in Mexico, we actually commissioned a series of flights, and so we are looking forward to work with this data. Thank you.
Secretaría de Medio Ambiente y Recursos Naturales.